Well, welcome everyone to our webinar series co-hosted by the International Oak Society and Global Conservation Consortium for Oak. This is our second webinar of our series. We had the first one a couple of months ago. So this is just really exciting to do again. We have a number of wonderful presentations in store for all of you today. So Andrew Hipp, Maricela, Rodríguez, and Alan Coombes are presenters today. But we'll start off with some announcements. Roderick will lead that, and then we may have some time at the end for some discussion. But just a few reminders, please stay muted throughout while people are talking. We will have designated Q&A sessions following each presentation. So you'll have an opportunity to unmute then to ask questions or make comments. But please feel free to put questions or comments in the chat throughout. And feel free to say hi and let us know where you're tuning in from. Thank you all so much again for joining. This is just very exciting to do. But we have a lot in store today, so I think we'll just go ahead and get started. And I'll pass it off to you, Roderick, for our announcements.
Well, thanks very much, Amy. Great to see everyone. I know most people here. It's great to see all these names. My name is Roderick Cameron, currently the president of the IOS. So welcome to our second webinar. I'm just going to go over some announcements regarding events. That's what's next on our schedule. I'm sure we're all excited about. But starting from furthest in the future, I think most of you may be aware that we've announced the dates for the next conference, which is taking place in Oaxaca in Mexico. That's from the 3rd of October to the 6th, 2025. That's October 2025. And as usual, we'll have a pre-conference tour and a post-conference tour. So the first tour will start on the 26th of September, extended to the 2nd of October. And then after the conference, from the 7th of October to the 12th, is the post-conference tour. The pre-conference tour begins in Morelia, goes down to Oaxaca, and then the post-conference tour will probably start and end in Oaxaca. So that's about 17 days you can dedicate to oaks in October 2025. Now, in the shorter term, the tour committee is working on the events for 2024. We're hoping to have 2 oak open day events in Europe, one in the UK and another in France. We're still working on that. This will be in May or June, and we hope to announce the dates and the venues shortly. But we hope to have those 2 events in Europe. And then in fall, Northern Hemisphere fall, of course, we're working on 3 different locations for tours. California, we're working with Stu Winchester, who organized or led our pre-conference tour in 2018. That may be in September. Then a tour in Taiwan, which would be very exciting if we can get that to work. We've already got our Taiwanese members working on organization for that. That's looking towards the second half of October. And then we're also talking to Adam Black about a tour of the Carolinas. This would be gardens and also field trips in North and South Carolina, and that is scheduled for around November. So it looks like a very busy time for us next year. This is a sneak preview for this webinar. We haven't announced this yet on our events, but hopefully, I'm hoping by around mid-December, when we send out the next issue of the Cupule, we'll have those more details on that. And finally, we will have our 3rd webinar, hopefully in February 2024. We still haven't worked out the details yet, but we'll announce that as soon as we have. And I think that covers the announcements for now, Amy. So over to you to continue. And just before I finish, at the end of the 2 presentations, we'll open this up to the floor for a general discussion. So any comments on the event schedule we can keep for then. Thank you.
Great, thank you so much, Roderick. Lots coming up for the iOS, which is very exciting. And yeah, we'll share the recording following this, as well as I think we can share a little blurb about these announcements that Roderick just shared as well, just for all of your information. But yeah, I'm happy to now pass it off to Andrew Hipp, who will be presenting on oak hybridization. Just to mention again, feel free to put questions or comments in the chat while Andrew is presenting, and then we'll have time for people to unmute following his talk to ask questions. So whenever you're ready, Andrew, thank you so much.
Thank you very much, Amy. And Roderick and Amy, thank you so much for inviting me to speak. Thank you, everybody, for coming today. It's really an honor to speak to you all and share some of this work we've got going on. So I'm going to try and share my screen. Share my screen, and I project my slides. I hide this doohickey. Are you seeing my slide? Yep. Fantastic. So what I'm going to talk with you about today is I'll give a general overview of some aspects of hybridization in oaks, and then I'm going to talk about a collaborative project I've got going on with a big group of colleagues that we're very excited about. I know some of them are in the audience. Alan Whittemore is here. Paul Manos may be as well. There are a few iOS folks in this collaboration that I'll be mentioning. If it looks from time to time like I'm looking down here, I have a clock on. I'm trying to keep myself to 25 minutes. So if I don't, Amy, you can start shouting and hollering because I won't see anything. I've got it set so I shouldn't be able to see things. Anyway, so oaks, hybridization is, of course, a deal in oaks. We know that oaks form good species. And by good species, I mean what Darwin meant by good species, which is entities that we can recognize across their range. You know, when Darwin was writing The Origin of Species, he didn't really talk a whole lot about what he meant by species, but later in The Descent of Man and in his notebooks he did. And he said, we know what species are, right? They're organisms, groups of organisms that when they have the potential to mix and merge, they don't. They're ecologically, morphologically, and that's as far as he was able to go at that time, distinct from one another. What he spoke against was the idea that species are strictly defined by reproduction. Because we know, like with oaks, there is interbreeding between oaks. Despite that fact, they maintain good species distinctions. So this is a nice figure from Rushton's work from 1993 showing that there is reproductive isolation between Quercus robur and Quercus petrea, such that the individuals that are intermediate have decreased pollen viability. In fact, there's been wonderful work on robur and petrea coming from Antoine's group. I see you in the audience, Antoine, showing this cascade of reproductive isolation mechanisms, but we don't have a lot of studies like that for a lot of kinds of species, and I suspect that it's going to vary from species to species. Anyway, at this rate, I won't get done today, so let me try and pick it up a little bit. So I think one of the things that we know with oaks too, across the tree of life, is that hybrids are more than a novelty. First generation hybrids act as a potential genetic conduit between species. So here we've got Tucker's some of Tucker's really beautiful work. Here looking at Gambellii and Arizonica. Here's Gambellii up in this corner. He's just mapping a couple of traits that you can use to identify these things, how lobed the leaves are and leaf width. When he looks at these two traits together, you can distinguish Gambellii, you can distinguish Arizonica, and then you see these things that are hybrids. See these little whiskers? He's using Edgar Anderson's method of ideographs for looking at segregation of individual traits. If you're interested in looking at some of that literature, it's Anderson's work is wonderful on this. But these first generation hybrids aren't where it stops. They offer an opportunity for genes to flow between species. So here now we have work looking at two of the North American white oaks, the good species, and then the intermediates. It's those back crosses that allow genes to move from species A to species B. And that movement of genes from one species into another, that's called introgression or introgressive hybridization, a term that Anderson introduced in 1938. Hijacking, as we often do, an old term that just meant it moving in this case, referring specifically to genes. One of the really great early studies was by G. Ledyard Stabbins, who looked at Quercus marilandica over here, Quercus ilicifolia. He got a group of graduate students from Columbia University, or at least advanced botany students who were working with one of his colleagues. And they went out to herbarium collections of these two species, to a pure site of Quercus marilandica, and then to a mixed site of Quercus marilandica and ilicifolia. And what they documented there is that it appeared, barring any possible biases in the data, and there are good reasons that these data could be somewhat biased, that in sympatry, the species were more similar to one another. They were drawn towards each other. And this is the power of introgression, to move species in ecological space towards one another, to share ecological solutions that have been innovated in one species and that may work well in another. Introgression can increase the variation of species and thus evolvability of populations. It can directly move ecological solutions between species, and it can play a central role in migration. So just a few examples. So for instance, when we look at Quercus gambellii, this weirdo here in the southern Rockies with this sort of bizarre distribution out here where there aren't a whole lot of white oaks, though of course the things that it mixes up with as the gelata complex, its peculiar range may reflect a history of gene flow between Quercus lobata, the species over here, and Quercus macrocarpa. And in fact, when you look at the molecular data, there's good evidence that both of those genomes are intermixed within gambellii. And its peculiar range combined with that fact is certainly suggestive that the genes that get mixed up in it allow it to survive in this area of the Rockies. Isolation of Engelmann oak in Southern California from a Northern Mexican ancestor, of course, Quercus oblongifolia, may have been facilitated by alleles gained from native Californian white oaks. Quercus engelmannii, located right down here, originated most likely from Quercus oblongifolia that came up along the Sierra Madre Occidental and spread out by the late Miocene, about 5.3 million years ago. It appears that Quercus engelmannii was in this area. And then as oblongifolia receded again, engelmannii got separated out over here. But it did gain genes from Quercus berberitifolia, one of the California white oaks. I wouldn't take this timing with a grain of salt. This gene flow may have been somewhat younger than this, but again, the success of Engelmannii is a very restricted species right now, but it is a Mexican oak lingering there in Southern California, perhaps due to adaptive introgression. And some of their unpublished data that's in O'Donnell's thesis, who's the primary author on the second paper, looks at some of the genes that are involved. Finally, this is work that you're probably all familiar with from Rémy Petit and Antoine Kremer et al., demonstrating that Cecil oak appears to have migrated northward across Europe following recession of the glaciers. By Quercus robur lobbing its acorns out, it disperses readily by acorns, pioneer species, and then being swamped by pollen dispersal from Quercus petrea. Transformation of a Quercus robur stand into Quercus petrea, largely facilitated by the very strong asymmetrical success of Quercus petrea pollen. I see Janine Cavender-Barris is on. Good to see you, Janine. Anyways, so these are all examples of gene flow potentially influencing the success of species. But not all genes need to move to shape species coherence. So our group is interested in understanding the genomic mosaic. Which genes are moving, which genes aren't, and how they facilitate success of species. Now, this is a collaboration with a lot of groups, International Oak Society members that are in this grant that I'm about to talk about. myself, Paul Manos, Ellen Whittemore, Janine Kavner-Barris, and Ian Pierce. Maybe others from the iOS in this group. I'm not 100% sure, but for sure, the 5 of us. This is a collaboration. I'm not sure if I can delete this, but anyways, this is a collaboration between several U.S. institutions from the Morton Arboretum, as well as Duke University, University of Minnesota, National Phenology Network, Fort Collins Science Center, and then 2 institutions from China, the Chinese Academy of Sciences and South China Botanical Garden. I'm going to pause this for a second and see if there's a way to get rid of these annotations. Amy, do you know how to get rid of the annotations by any chance?
I'm not sure. So maybe going up
I'm
in
going to
the
disable
control
annotation at least.
Yeah.
Looks like there's been something going on there. Well, we'll keep the annotations on there and they'll make it that much more interesting. Okay, so continue on. I want to acknowledge that this is work that is being conducted by a lot of people, including many students, postdocs, both graduate students and undergraduates, and postdocs, as well as our research assistants in the labs.
Hey, Andrew, if you go to your annotation panel, there should be an eraser function. So like at the top of your screen, if you hover at the top of your screen, you should be able to see a panel, the control panel for you to erase.
Oh, there we go. There's an eraser. Oh, thank you so much.
Yeah, thank you, Max, for mentioning that.
Yeah, thank you guys very much. I appreciate it. Okay. So, and if anybody wants to jump in and ask questions, please do. If there's anything you want me to point out on here, I'm more than happy to do so. So the grant we're undertaking investigates how phylogenetic history, genetic diversity, introgression, and functional traits influence one another, as well as the communities of organisms that live on oaks. So we're really asking about how this gene flow shapes the entire forest community. The project is 3 projects at once. One is looking at genomes, genetic diversity, and phylogenetic diversity in natural systems. Symbionts in natural systems are looking at fungi and insects in natural systems, and then using a set of common gardens and biodiversity ecosystem function experiments to look at, again, look at the effects of gene flow on oak diversity and the diversity of its symbionts. All these projects are run in parallel in China and North America. Today, I'm just going to be focusing on the genetic and genomic questions. So what oaks did we select? We're focused on bur oak and relatives, partly because they're a classic group for studying introgression in Eastern North America. You can find intermediates between Macrocarpa and numerous species in Eastern North America. This is a collection by Mike Nee, where at his family farm, he found Quercus alba growing, Quercus macrocarpa growing, and between them intermediates. This is a group that had been studied really extensively by Baranski and Hardin in the 1970s. James Hardin wrote a really foundational paper in 1975, arguing that while most of the variation in Eastern white oak Quercus alba was intrinsic, genetic variation within the species, there was plenty of gene flow between species as well. So here you've got 16 white oak species of Eastern North America, each line indicating a hybrid that had been observed in the wild, either experimentally or from herbarium specimens. This group was then revisited by Bill Berger the same year, who pointed out that there was so much hybridization in the group that you couldn't really take the biological species concept, this idea that species are defined by reproductive isolation. If you did, there would really be only a few biological species in North America, because it seems that all the white oaks can potentially interbreed. All the red oaks and relatives can potentially interbreed. So he said we need something better. Lee Van Valen, then working at University of Chicago, the next year comes along and says, well, let's use oaks as a group to sort of probe this idea of what species are. And in his view, what defines species was their ecological niche. He says species are defined by having a unique position in the ecosystem, a niche that involves their interactions with other species, their relationship to the abiotic environment as well. And it also involves some genetic interconnection, whether that's by gene flow or by ancestry, was irrelevant in his mind. So, and there's a great quote from his paper in 1976, indicating, just arguing that it might well be that bur oak in Quebec exchanges more genes with local swamp white oak than it does with bur oak in Texas. And his point here being that it's not gene flow within the species or reproductive isolation between species that define species, but rather their niche. This was looked at most closely first by Alan Whittemore, who's in the audience now, in a 1991 paper, where he and Barbara Shull, he went out and collected specimens of Quercus alba, Macrocarpa, Michoii, Stellata, virginiana from throughout the range, throughout much of eastern North America. And what he discovered is that the chloroplast DNA is shared not based on what species he collected, but what site he collected at. He found multiple chloroplast haplotypes or genotypes at each site, and the dominant one at each site was shared amongst those species at that site. By contrast, however, the nuclear data showed a discrimination of species. Quercus stellata, post oak, was distinct from both Quercus alba and Macrocarpa, eastern white oak, and bur oak. And while it wasn't a ton of data, it did suggest that the nuclear genome was telling one story, the chloroplast another. So as a group, many of us who were in this collaboration went in to investigate 7 species from Hardin's 1975 Oaks and Gamion paper. We wanted to get some genetic data to ask the question of whether or not these species are genetically coherent across their range when you look at all of them together. So Mira Garner, who was working in our lab at that time, undertook a big collecting trip across much of eastern North America, in addition to which Janine Cavanagh-Barris collected these samples from the Black Hills. We had Paul Guger collect these samples from out east, and we had some other samples as well from Guy Sternberg's common garden of bur oaks down at Star Hill Forest. A postdoc working in my lab, Elizabeth Fitzek, went to Piraton, worked with Erwan Guichou, and developed a mass array SNP set for 80 SNPs initially, 75 that ended up being useful. What it was a gene typing toolkit that subsetted a very small amount of the genome, just 20 variable nucleotide positions, the A's, C's, T's, and G's that make DNA, that suffice to distinguish this set of 7 species. When we look at them and we cluster these data, what we find is that these 7 species can be defined by just a very small number of regions of the genome. You'll notice these long branches. You may look, if you're used to looking at these data, you may say, well, that's a lot of genetic variation leading to those species. This is a biased subset of SNPs. They were selected specifically for variation between species, low variability within species. But the question is really not what is the absolute rate of admixture or anything like that, but can we recognize species across their range? And genomically, we definitely can. Yet interestingly, despite the fact what you're looking at here, each column in this figure represents one individual. The color combinations in that column represent the portion of that individual's genome that's estimated to have come from one of, in this case, 8 ancestral populations. So this individual here looks like it's a hybrid between Muhlenbergii prinoides, we can't distinguish those molecularly, and Stellata, for instance. So despite the fact that we'd chosen markers that should be invariant within species, we still found evidence of integration. And this suggests that the Eastern North American white oaks are a true syngamion, a term that was coined by Lotze in 1917, and just means an interbreeding group of species. What Lotze's view of species was at all is a little unclear to me still. But this term syngamion caught on. And the way we use it today is to refer to a group of species that we think of as good species that nonetheless swap genes fairly openly. So we wanted more than just the aggregate story, right? We wanted not just to know what's the total story, this sort of, here's Gambellii arising from Macrocarpa and Lobata, maybe, but we wanted to know what's going on with each gene, right? So here's a story that we had sort of taken some stories and dissected them for our 2020 paper, some introgression histories, and found that introgression in one lineage of the white oaks tree is not always reflected by introgression in another area of the white oaks tree. So maybe different genes recording different histories in different areas. So to try and get at this, we developed what's called a HybSeq bait kit. This is work by Casey Pham, who's been working in our lab on and off since 2013 when she was a high schooler. She's now a graduate student with the Soltis lab and still collaborates with us. Bless her. She developed a kit for 465 genes that we could readily fish out of the genome. Here she is metaphorically fishing, and sequence these genes fairly efficiently. Our method was we took these 465 target genes and we sequenced them across 517 individuals. Again, individuals that have been collected by Mira, who's gone on to a permanent position at the Field Museum just this past month. We miss her dearly, but we're happy that she found a good permanent position. And to take a look at what the introgression patterns are. So analysis was conducted by Gabe Ribikoff, an undergraduate in my lab. In the end, for those 465 genes, pruned down to 445 that worked well, you got a total of 29,000 SNPs. To those of you in the audience who are like, well, that math doesn't work, Andrew. What's the sort of what linkage is there between those SNPs? I won't show the analysis, but suffice it to say that when you prune this down to a single SNP per gene, you get essentially the same results. So the exciting findings are, first of all, okay, these species are good species, right? There's Quercus bicolor. Here we're looking at an ordination. We've taken all the genetic data and we're just representing it in 2 dimensions. Where the distance a dot is on the map, on this plot, indicates its genetic distance from other individuals. So all the Quercus bicolors are fairly closely related. All the Quercus macrocarpas, the bur oaks, are fairly closely related. But there are individuals that seem to fall halfway between the species. And when you look at the data using, again, this method that we looked at a few minutes ago, where we're asking what proportion of each individual's genome is attributable to each of a set of ancestral populations, either 5 or 6 in this case. What we find is that, first of all, there's nice, strong genetic structure within Quercus macrocarpa. We'll come back to this in a moment. There are numerous individuals that are F1s, first generation hybrids. And these, we double checked. We used a second method called new hybrids that allows us to fit a model. We say, is this an F1 or is it a later generation hybrid? The ones that look like F1s are F1s with high probability. And then a little level of detectable gene flow that goes beyond the first, second, or third generation of crossing, which is interesting. I expected to find just a smear of variation. The variation within Quercus macrocarpa, pretty interesting, seems to have strong geographic structure. This is an analysis called FEAMS that's developed by John Novembre. All this was run by Kieran Althouse, who I saw is also in the audience. Thank you, Kieran. What this shows is is a barrier to sort of gene flow between the north and the south, approximately, suggesting that there's a sort of a strong geographic structure like we see on the map. This may have an ecological component because some of this genotype may be associated with bottomland forests. This is something that Gabe Ribikoff is investigating now, and he's hoping for answers by the end of this week. Second was these high proportion of apparent F1s, which again really surprised me. What I was expecting to find is very few F1s acting as conduits for a sort of long smear of variation where lots and lots of individuals would have low levels of admixture from multiple species. We're going into this in greater detail because this is just scratching the surface here, and I'm almost out of time. But briefly, we now have genome resequencing data for about 700 individuals. This is whole genomes. Sequenced at just 20x coverage, so that you've got a layer of sequencing coverage across the entire roughly 35,000 genes in the oak genome and everybody in between. This is data that's being analyzed by Rebecca Mohn, who's a postdoc in our lab. Initial findings, she's just focused on the chloroplast now because she was able to rapidly get that out. She's now analyzing the nuclear data. But sort of going back to Ellen Whittemore's work of 1991 and asking what the biogeography of the chloroplast is on the landscape. And you remember how Ellen's work showed that the chloroplasts didn't segregate by species. What you see here, these are all different chloroplast haplotypes. Look at the numbers of species that are within each of these chloroplast haplotypes. Pretty clearly, chloroplasts are shared among species. And when you look at the geography on the map, it looks a little bit like a spider web or a train wreck. Think of it as you will. There's tons of shuffling. If you've looked at Antoine, Remy, and the other Europeans' work on this, on the chloroplast variation, what you've seen is this beautiful story where they have 3 refugia that moved northward and began to intermix as the glaciers receded. What appears is happening in Eastern North America is that everybody was together during the last glacial maximum, and there wasn't a chance for this clean sorting, with some exceptions. Black Hills certainly sort out nicely. Anyways, coming to the end, we're going to be using these data in combination with a set of reciprocal transplant experiments that Janine Cavender-Bears, who I believe is in the audience, designed and really has been dreaming about this project for a long time and got it going in 2021 before our funding came through. So thank you, Janine, for making it happen. This is the 2021 design and implementation team. This is just the Morton Arboretum garden. This is one of 3. One at the Morton Arboretum, one at Minnesota, one at Oklahoma. Here we are getting seedlings that Janine and her crew had gathered all the acorns that we collected at our individual sites, had them planted out at Valonia in their tree nursery. And then we went down to Valonia, picked them up, and laid them out, planted them out. And here's what it looked like with a lot of help. There's Lucy. Here's what it looked like at the end of 2021 with Oklahoma and Illinois trees already looking distinct from each other. And here it is in 2023, the same plot after we planted the second half, which is in the back of this photo. You can't see it. And some of our trees planted from acorn in 2019 are already producing acorns. Looking forward, we're going to be using these data also to investigate the gall wasp specialization and the endophytic fungi within the leaves, as well as endophytic fungi in the roots, as well as at Minnesota, looking at Janine's experiment, ecosystem biodiversity function, a biodiversity ecosystem function experiment, looking at the community effects, to ask this broader question of how oak diversity shapes our entire forest and savannas. In addition to all the collaborators, this kind of work doesn't happen without a lot of help at home. And I owe all the stuff that is happening back at the Arboretum really happens because of my lab's support and institutional support. Okay, thank you. I'll turn off now and I'll entertain questions if we have time. And if not, that's okay too, because I think I used up all my time.
Yeah, no, wonderful presentation, Andrew. Thank you so much for sharing that incredible work, the incredible collaborative work that is ongoing and has been done. We do have, we have about 5 minutes. I think we're good. We'll be okay for questions. Maricela put a question in the chat asking about They have hybrids between species in different subgenera, but no hybrids between Cyclobelenopsis and other Ceres species, or hybrids between Quercus and Labate. Why is this?
Great question. I don't know that we have a good understanding of why sort of Labate and Cyclobelenopsis seem to really hold themselves off. I don't have information on that. I wonder if is there any chance Min Deng is on the call?
I don't think so.
Okay,
No.
because Min has done the work that I suspect will get us closest to an answer on this. She's done that really meticulous pollen tube work where she's done some manipulations and tracked what happens with the pollen tubes. If there's anybody else in the crowd that does have insight into those particular kinds of incompatibilities, I don't know.
You can say something.
That's my question.
Do you want to follow up with that, Maricela or Alan?
It's my question actually, and I didn't really think there'd be much an answer to that, but I just put it anyway because it's always interested me which oaks actually hybridize and which do not.
One thing we do know is that the genome structure is very consistent across. I mean, you find micro rearrangements, you find some larger rearrangements, but it seems like as you cross the entire genus, so it's that fixed, that really stable genome structure is really striking. So it seems like they're all predisposed to pair well at meiosis. But then there is something restricting crossing between at least some of the subgenera. I don't know, sections. I don't know what it is.
Great, thank you. And then
Roger.
Roger, I see you have your hand up, but Andy put a question in the chat first, so we'll go to that and then to you, Roger. So Andy was asking about when collecting acorns in nature from a specific oak species, how many trees need to be present within a population in order for the collection to have a high probability of representing true to type genetics for that species being targeted?
That's wonderful. And we have expertise in the crowd. I know Austin is on this call. I don't know if Sean is, but Sean Hoban's lab does the leading work on this exact question for not just for oaks, but for trees in general. Austin, would you mind responding to that? Because I think you have the best expertise in the house right now.
Sure. I can't give a hard number because it depends really on how many, members of this syngamion or whatever network you're looking at. Obviously, the more you have, the more representatives per species you need. I think in like systems of 4 or so acting individuals, you need a minimum of like 5 true to type. But there is a lot of interaction there between, how many back crosses versus F1s you have. So if you're interested, Andy, I can direct you to some papers that our group and others have done on that particular question.
Wonderful. Thank you so much, Austin. And thanks, Andrew. Did you want to add anything to that? Sorry.
No, I just saw that Roderick had his hand raised.
Yep,
Thank you very much, Austin.
Yeah, my question is, I've come across in the literature the concept of a stabilized hybrid. Like certain species say, well, this is a stabilized hybrid. I'm not sure what that means exactly, and it'd be great if you could tell us. And also, isn't every species a stabilized hybrid?
It's a great question. If you look back far enough, probably we know that there are ancient genome duplication events, whole genome duplication events, and some of those will have involved hybridization. Like all of the Juglundaceae is argued to be of hybrid origin. So that would be where you get 2 species hybridizing, and then that hybrid becomes reproductively isolated from both parents and goes forward and diversifies. So the idea of a stabilized hybrid, like when you look at some of the sunflower work that Lauren Reesberg's lab did, what they argue is that you have species 1, species 2 mixing, and they document, for instance, in North America, they found 3 species that looked like they were stabilized hybrids arising from crosses between 2 species. They then become reproductively isolated from the parents and go forward and have a trajectory of their own. So that's the idea of a stabilized hybrid. How often does that happen within oaks? It's not clear to me. So for instance, when we look at Gambellii, those data are really compatible with saying, oh, Gambellii is a stabilized hybrid. Or you could say Gambellii arose from the California white oaks and has had such extensive introgression from bur oak that it sort of got tugged over this direction. When you get high enough, the clear-cut examples are polyploid speciation, where you get species 1, species 2, and then duplication of the genome, and that individual can then be reproductively isolated from both parents. That's sort of instantaneous. We don't see that in oaks for sure.
Thank you.
Yeah, Janet, the question about nomenclature, I'm going to defer to Alan Coombes, if you'd like to, or Alan Whittemore, since there was a nomenclatural question.
Sorry, what was the question?
The question Janet asked is basically, she said, talking about named hybrids like Quercus ex McDonaldii, and she was asking who controls those names. She's asking if there's an organization that names them or controls names.
Well, it's everything is a matter of opinion. One person can regard something as a species, another person can regard it as a hybrid. I don't think there's any rule. You just have to make a decision.
Oh, Janet, I'm sorry, that was a direct message, Janet. Yes, exactly. And so there's not a governing authority, but there is a standard of assigning names, and the standard for giving names to hybrids is comparable to the standard for publishing any name, is it not? You have
exactly
to get
the same.
You have to get it through review. So yeah, there's not a sense. Oh, sorry.
Sorry, I was going to say one thing this does have is implications for conservation, of course. You know, if you're conserving a species, you don't want to go around and collect just what is exactly that species. You need to collect all the little bits of it that have got other species in as well.
All right. Well, thank you so much for this discussion and these questions. And I see Alan made a comment too in the chat, so be sure to check that out. But I think we need to move on to our next presentation, but please feel free to put other questions in the chat for Andrew throughout. And obviously for Maricela and Alan, who are our next presenters. But yeah, thank you so much again, Andrew. Really appreciate you taking the time and sharing this incredible work. But I'm very excited now to pass it off to Maricela Rodríguez and Alan Coombes, who will be presenting on the oak collections at BUAP. So whenever you two are ready. Thank you.
Thank you very much to Roderick and you for organizing this event. And we are happy to participate here like a team. And we will join and we will be together in the presentation. So I hope you enjoy. It's something very simple, not as complicated like Andrew, but I hope you enjoy. So I'm going to share my screen. Is that okay?
Yep, just in full screen.
Yeah. Okay.
Perfect.
So what you are seeing is view of the botanic garden when it's pretty and it's rain. And we call this presentation the oak collection in the botanic garden in Puebla and the university. For those who doesn't know where Puebla is it's in Mexico and is very close to the volcanoes. So you saw in the promotion poster one of my pictures in the last before they built a huge tower for the rectoria at the university, and now you can not see that view anymore. So I think it's a very pretty view of the past. So in this presentation also we link the collection we had built toward a meta collection. So I think not everybody is familiar with this, but we will try to explain a little bit about this. So we divided the presentation in 4 little parts. And the first one, I want to talk a little bit about the origin of the Quercus collection in the Botanic Garden at the university. And later, we will talk about how this collection was developed. So further, we will talk about how this previous collection is helping us to create the national meta collection. And at the end, we will finish with the future of the collection. So I'm going to start because time is running, but I think some people knows about a little bit about the origin of the oak collection in Puebla Botanic Garden. But I want to give you just a few background of this. The National Collection were an initiative of the Mexican Association of Botanic Gardens, and that was in 1992. This wasn't a new and an original idea of the association. It was a copy of what was launched in 1982 with the foundation of the National Council for Conservation of Plants in Gardens. So we got carried away in the association and we asked why we shouldn't have national collections in the Botanic Gardens. And we started to discuss what this collection should include. So we were very good at setting standards. And we say, well, we should have at least half of the majority of the taxa in the group. And I thought, what happened if we want to have the Asteraceae family, for example? Yeah. And then we said, well, it should be created product of research or for it use or for educative purposes, and we must include a specialist in the group. This was very high hopes because even now in the Botanic Gardens, we don't have a specialist botanist in the garden. So, but we like to dream. And then we realized that the only collection we could have in that moment that it would be like the national collection, it was agave, not trees at all. So I was very brave and I said, well, Puebla has 10 hectares garden and we can have a national collection of trees. And my favorite tree, it was the oak. And also in Puebla, there is a lot of forest. So I thought it was a very, very good idea. So we started to work in the oak collection, and curiously, it didn't start at the university. It started in a very small botanic garden inside a zoological park. And I wanted to give the name of the owner's mother, Luis Worley de Camacho Botanic Garden. And our first important visitors were Alan, Guy, Susanna, Kevin, Mark, and me. Of course, I am behind the camera, so you cannot see me, yeah? But I want that you know that I was there. So they visit the garden, the small collection we have there, and also in that time, we had a very big nursery where we were propagating 200,000 oaks that they were going to be used for restoration. So from 1995, the Botanic Garden started to help at the university, started to help the duplicate of that collection in the Luis Worley de Camacho Garden. And by 1999, we reached 75 species, but few of them were planted. So it was very sad to see that the collection failed after this. Why? For many reasons, but most of the oaks were in containers, very small. Some of them did not grow well. Some of them died as soon as they were planted. And there was a very huge horticultural failure. So in addition to this, the zoological park started to have financial problems, and my dream of having land and money for this collection failed. So it was until 2007 that we started again to recovering the process in the collection in the university. What happened then? Well, the new development of the collection started in 2009 when Alan Coombes started the curation of herbarium, BUAP, that's the name. Then realized that he had to curate also the living collection. Then we realized that we needed all to know all the flora in the Puebla state, and we spent from 2010 to 2014 in elaboration of 3 key books to identify the plants in the wild for all the Puebla state. In every field trip we did, we included oaks. So that's why we were starting again the collection, and it showed an improvement. So who is better to talk about the collection than Alan? So I'll leave Alan. Your turn.
Thank you, Maricela. We'll move on to looking at some of our oaks in the garden. This is our garden, an aerial view. It's obviously in the dry season because otherwise it would be lush and green. Not only, that's not our only collection, because in recent years we've developed 3 external sites. Firstly, because we don't have a lot of room, only 10 hectares here. Also, our climate is pretty rough, so we can go for 5 months maybe without any rain at all. So oaks have to be tough to live in this site in Puebla, and they actually are very tough. Next. Okay, this is where our sites are. In the middle, that's the garden in Puebla. So you see we're pretty far south in Mexico. We've had the 2 collections in the north of Puebla there, which are in places much more humid than we are. And another site is we have some oaks in Acapulco Botanic Garden. We're still waiting to hear what's happened to those because the garden has recently been devastated by Hurricane Otis, and we have a team there now trying to help clear up. Yep. That's a list of everything we have. I can't leave it there for very long, so we don't have a lot of time, but that's the list of all the oaks we have at the moment. Changes every year because normally every year we're going to plant something new. We're going to do the aliens first, the non-Mexican species that we have here. We do have a few, starting off with some Mediterranean ones. Quercus ilex grows really, really well. You can see that's starting to be a very nice tree. These Mediterranean ones are ones we collected in southern Spain about 2000, I think, but it's growing a lot better than some of the Mexican oaks there. And also growing well is Quercus rotundifolia, making a really nice tree and really different to Quercus ilex. Next, we're going to go through these aliens pretty quickly. Quercus suber, it was also one of our collections, but that tree on the left, obviously not pruned properly when it was quite young, and so has developed a flat top. But the next one is more recent planting of probably the same collection that I made sure is pruned up, so it's going to make a decent tree eventually. And Quercus ceres on the right, not terribly exciting and not growing very fast, but there it is. Next. Quercus robo, like me, suffers in the extreme heat we get, and you can see it looks pretty ropey there. Actually, this was in October, and it's still got some of the old leaves on, and the new leaves are coming out, and it's starting to flower. So I've seen better ones in a cooler garden, but here it really doesn't like it very much. And then we get these surprises. This is one that's slowly started growing and is growing quite well now. We received it as Quercus patria. and it was from acorns left at the conference in 2009. So I've no idea. This, I mean, to me, it looks very like a fastigiate roba. The petiole is maybe a little bit long, but apart from that, it's very close to roba. But if anybody who's watching this remembers leaving a seed of Quercus petrea at the conference, please let me know, because I want to know where this came from. But it's possibly the only fastigiate English oak in Mexico. Next. Of the Americans, we have Quercus buckley and Quercus schumardii. Potentially, we could get Quercus sternbergii, couldn't we? But that's maybe a little unlikely. On to the Mexican species, and we have oaks not only from Puebla, but from pretty much all over Mexico. That's a nice Quercus microphila, and this fruit's very early that's in July, when the acorns are practically ripe. That's a shrubby one, of course. Next. We have a big conservation collection of Quercus brandigi. All came from wild collected seeds from Baja California Sur. And the first photo is when we planted our first one. And there's Susanna, Maricela, and me looking over the young plant very proudly. And today, the largest one has grown to about 2 meters tall. That one's growing quite well. They're not all as good as that. Some have not developed leaves yet, so they really need some careful pruning to get them to grow up into a tree. But there's about 25 trees there. Quercus rugosa, of course, does very well here, although it comes from a lot higher than we are normally. It's a tree of the mountains, and flowers very well. And there I managed to find a photo of a bee feeding on the male flowers there. Yeah. Of course, this does do some strange things sometimes, and a lot of oaks do occasionally produce something weird. So I found recently these deeply lobed leaves on a tree of Quercus rugosa. Why this happened, I don't know, but it's still there, and it seems to alternate in the first and second growth. One stage is lobed, the next stage is normal leaves. You can see that on the leaf on the bottom left. Whether it could be propagated by grafting, I don't know, but it's certainly something very strange. Some people may have read about this in one of our notes. There was a big confusion between Quercus lancifolia and other species, and we had trees from different places that looked completely different. So I looked at them with Susanna, and we decided they represented different species. So this is Quercus lancifolia. Some of these trees may have been called Quercus laeophylla at one time. Lovely red new growth and very glossy leaves. Surprisingly hardy, because this came from quite a humid tropical place, but it's doing very well even in the very dry conditions there. And that's the Quercus toxicodendrifolia. Different thing altogether. It looks completely different. Very dull green leaves. This can have the young growth green or sometimes red. We've got the two different forms, but that one's growing very well. Quercus insignis, of course, is something that everybody wants to be able to grow, but very few people can. We saw a very nice plant at Tregrayan this year, which still had the purple young leaves even when they had matured. We have an older plant of this, which was put in a dry place, and that's growing, but not very fast. This one I put in a place that I think gets more humidity and not so much direct sun, and it's growing really well. You see that's growing very fast now. So we don't give up on this. It does come from very humid places, but what it wants more than anything is heat, probably in the summer, to ripen the wood. But that's going to make a very nice tree. How many people have been asked what's their favorite oak? Well, probably most of you. And how many people can decide what it is? Well, I change all the time. You know, you can't. You have one species of favorite, and then it changes to another one. This one's coming up to very close on my list now, which is Quercus cibifera. And I remember this species for one good reason, is because when I first saw it was on a field trip after the conference with Piers Trehane. No acorns then, but we went back the next year and collected them, and now we have several trees. It's a small tree or a large shrub. It's never going to get very big. It's lovely blue foliage, peeling bark. You can see that one there is starting to sucker from the base. And this lovely blue foliage and acorns. At one time I thought this was a separate species, because when I first saw it, the young growth was completely glabrous, and Quercus cibifera is described as being very hairy young leaves that you can see on the type specimen. And then I noticed the second growth on this one is covered in hairs. So I think it is the same as the one described from Chiapas. I think this is in Europe, but maybe not growing as well as it is here. So it's really a very nice tree. And look at the beautiful color of those leaves. Next, for an all-rounder from Mexico, I'm going to suggest Quercus gregi, because it's very tough. I think it will grow just about anywhere, and it's very, very variable. Here we've got the first four are the tree and the foliage underneath. So we have a nice, fairly columnar, dense tree form there, and they always have this lovely white, densely tomentose undersides of the leaves. The next one I call dumpy, because it is dumpy, and it's making a little tree there with a short stem, very slow growing, but very nice broad leaves. And then we have on the right two different forms. The first one is collected from the north of Mexico, from a shrub only about 1 meter tall, and already the plants here are taller than that. The one underneath that, with the nice bronzy young leaves, was grown from seed collected from a large shrub with leaves with no teeth at all. So I think a lot of this variation is environmental, and the shrubby one is eventually going to be a tree, and obviously the one that had no teeth has obviously developed teeth all of a sudden. Very interesting species. It's a potential of a lot of selections from this species. A quick run through a few extras we have here, going from the top left. That's Quercus frutex, growing really well, a shrubby species. Next is Quercus invaginata, which came from Coahuila. The lovely bronze leaves there of Quercus konzati, which was the first time we found it in Puebla. It only grows in the far southwest, close to the border with Oaxaca. Then Quercus germana, which is very reliable for producing its very large acorns, can be almost egg-sized sometimes. You might be offered some seed of this from a garden, but I suspect it's going to be hybrids. But there's a great potential here for developing interesting selections by getting crosses between these evergreen Mexican species and hardy temperate species. Just a question of getting them to fruit in the right place. The one now on the bottom left, we have Quercus jalapensis, and this can be very similar to some other Mexican species, but you can tell it by its biennial acorns. And there you can see the acorns which developed from flowers of last year, as well as the flowers from this year, which will be acorns the next season. That shows you it's definitely a biennial. And the last one is Quercus deserticula, a very nice bronzy young leaves, but very slow growing at the moment here. Now do pay close attention to your oaks, because they can do some strange things. Recently been hearing about an oak that has 2 or 3 acorns in the same cup. This one, the Quercus on the left, we have Quercus grahami, and we noticed this gall growing on them. Now, we've been working with somebody who investigates Mexican oak galls, and he's shown that this is a new species, and it's going to be named after me. Well, I prefer a majestic oak, but a 2mm wasp is the next best thing, I guess. And the other one is a hybrid raised here, which was found a new species of powdery mildew, which terrible thing in some places, but very little studied in Mexico. This one has already been named. At the end, now please visit our herbarium website, where you can find all our herbarium collections. You can get images to help with identification, and you can get distribution maps. That's a distribution map of Quercus hertifolia. The specimen is Quercus corrugata. We haven't imaged all the oaks yet because we need money for a camera, because they don't scan very well. But it can be a very useful site if you want to help try identify things or see where things grow. That's the address there. And now back to Maricela.
Okay, so, well, you know now the collection existent in the Botanic Garden, and I must tell you that we have been working in it, but since 2016, the Morton Arboretum became more involved in oaks, and several actions and conservation projects were launched. So we started to work with Morton Arboretum in a project of conservation which involved the continual field exploration, and we basically focus in Oaxaca, Veracruz, Hidalgo, the states around Puebla. So you can see in the picture one of the meetings where we have been participating. Murphy is there, which has been the very, very strong research in oaks science. And Antonio, Susana is here. And later, a little later, in 2019, the BGCI launched the Global Conservation Consortia, and it was the way the GCCO started. In this picture, Andrew, who was working with Murphy, is handing the consortium to Silvia Alvarez-Clare, which is here, for those who doesn't know. And Alan is in the steering committee of the consortium, and I am in front of the Global Conservation Consortium for Oaks for Mexico and Central America. So what is the connection now with this collection and our projects? Well, the projects in Morton Arboretum is targeting priority species, and even when Puebla Garden had a good collection now, it had very little species threatened, and we focus on that. So distribution, cultivation, and conservation is basic for us, as fundamental also. And here you can see Quercus hirtifolia, which is an endangered species that Alan showed the map before, but he didn't tell you that I think this is the best map of distribution of any species in Mexico. So you can see here all the routes, and every time we are following the steps of this species, and it's incredibly interesting because we had find interesting genetics results. We are working with Dr. Antonio Gonzalez, and recently it was found for one of our collaborators, a very, very isolated rock where this species is. So we are really interested in know more about the genetic diversity of this species, and I think it could be a good model one. Here to the right, you can see the beginning of the meta collection. We commit many mistakes in the past, and now we want to grow better the oaks we have. So now that we are focusing in priority species, we try to keep more order, but also we have more problems with that because we don't have that much space and that much money to build a large collection. But that's a topic we need to talk later on. By now, I want to tell you here, I want to show you the species, the priority species we have here in this botanic garden. And you can see we have 11 now that they are in the endangered or the threatened plants list, written by IUCN. And you can see the ones involved, Brandeji, Delgaduana, Irtifolia, Insignis, Rubramenta, and Meavi, that we have in large numbers. We are trying to grow more individuals and more origins. If you see in the next column, you can see other species that also are part of the collection. They are 13, which are data deficient or near threatened. So in total now, the collection in the Botanic Garden, it has 62 species, plus 22 which are growing. So we can say now that is the national collection because we have the 50% of oak species, finally, and we have 11 of 32 threatened species. So I would say that what GCCO, led by Morton Arboretum, has helped is to improve, to increase the number and the quality of the collection in priority species. And I think this sets an example for other gardens. I want to show you two things that you oak lovers are going to like. Two priority species that we are working with. Quercus delgaduana, the one who is to the my left, and is growing many plants of these, not just in the Botanic Garden, also in Veracruz. And we did an agreement with people who work for the presidential program of planting in Mexico. And they have grown these plants in the nurseries they call Sembrando Vida. So we have thousands of these Quercus delgaduana growing in situ. And then I'm going to introduce you, maybe first time for all of you, to Quercus rubramenta. The one in the middle is the giant of Mexico. It's the tree that we want to see, 65 meters tall, and is living in Guerrero. To the right, you can see how it's growing. We have 300 in the Botanic Garden, and thanks to the iOS who funded this project. So, but we don't stop there. We have this majestic tree growing in situ. We have 3,000 seedlings that will be planted in situ. And of course, we need to work more with the distribution, but at least we started. So thank you to the iOS. I hope they carry on helping us. So finally, I want to talk about the GCCO and the steward training program. The experience tells us that we cannot carry on working alone. We need collaborations. So we need an army of people trained in oaks. And so we create the steward training program for Mexico and Central America. We have held 2 programs thanks to Amy, who is the leader of the GCCO. You can see her in the picture. And we organized two workshops, one in Ensenada, Baja California, and one in San Miguel Allende, and both were a success. So we not only train the people as a steward, if not, we got the attention for almost all the botanic gardens in Mexico now, which at the beginning, they weren't very kind to participate. So if we have the stewards are going to support the meta collection. That's the what we need, because none of the botanic gardens in Mexico at the moment have the facility or the capacity to hold a large oak collection. So we need to work in different way as you do in America or in other parts of the world. So if you can see in this map, most of the priority species are in the oriental mountains in the west, in the east, sorry. And you can see here, basically all the threatened species are in this area. However, thanks to the collaboration with the stewards and this program, we now have Guerrero and Baja California also included in our map. So we also carry on with the planned donation of some priority species to establish gardens to invite them as part of the collection program. So what is the future of the the collection? As I see as GCCO representative in America, in Mexico and Central America, I would say that we need to collect the 21 priority species remaining, and we would like to do it by next year. We want to involve the stewards or guardians in every state. We need them. Also, now that we see that the propagation is not exactly the problem, how are we going to get the land? That's now the problem, because our gardens don't have land. So we need to work in another way. We need to take care of the genetic composition. Irtifolia is a very clear case of it. We need to work more with Andrew and Antonio and other experts in the topic of genetics. We need to support and involve other botanic gardens that we are doing in Mexico and in other parts of the world, especially in America. And we need to promote the collaboration between different countries, especially the neighbors one. And I hope we continue our work with the Morton Arboretum, saving these beautiful oaks. As I mentioned, collaboration is the key. And here is a field trip we did. And I would say this is the first study trip we did. So it was full of experts devoted to focus in understanding the taxonomy, the complexity of the hybridization. And I hope we can do something else or more trips like this. And thank you very much for your attention. And if you want to contact me, you have my details there. And thanks very much to the GCCO for giving me this opportunity to work with this beautiful group of trees. And thanks to Alan, who is working still in the University of Puebla. Thank you.
Thank you so much, Maricela and Alan. Wonderful presentation. Any questions? Feel free to unmute or put them in the chat. Andy asked for the link for the herbarium. So if you could put that in the chat, Alan or Maricela.
I will send that to you, Amy, and you can post it to somewhere or to anybody who wants it.
Okay, sounds great. Thank you. other questions or comments for Alan or Maricela?
I have a
Yeah.
question. I was particularly pleased, you'll hate me for saying this, but to see you discussing your horticultural failures simply because it made me feel less bad about mine. What was it that led to success? I mean, what were you doing wrong that then worked later? For example, I noticed you had some very good mulch there. I couldn't tell what it was. What are you using for mulch?
Well, I would say that we inherit that, but there is no really interest in Mexico for horticulture. Gardening is in like something which is not important, and it's very difficult for people to understand what a botanic garden is. So we have to train the gardeners, but they are basically general workers. So there is no school. So that was one of the big problems that we have at the beginning. Nowadays, there is a better team, but still we see that horticulture is a real bottleneck. So I don't know if you are happy with it, but we really struggle with horticultural skills in Mexico.
Sure, thanks.
As an example, I can say when I started, I stopped the gardeners doing mulch for volcanoes and painting the bark white. That's the sort of state they were in. And I think a lot of the oaks failed because the pots they were in were too small. They planted them too small. The root systems were not big enough. So now everything is grown in big pots to have a really good root system, because that root system that needs to survive the dry winter after planting.
Well, they're looking great now.
Yep.
Philip was saying he's never heard of Quercus autopista. Could somebody provide more information on that taxon?
Yeah, this is a bit of a problem at the moment. Autopista is something we collected in 2008 on the Autopista in Oaxaca, and we grew it. And then the only place I've seen the two growing together, which is the one it's similar to, is Mignonensis, and that was in France in Beatrice's garden. And these two are identical. It's very difficult to tell them apart. And they come from small populations, 700 kilometers apart. I think there has been some genetic work recently on this, which was inconclusive, maybe, Andrew. I'm not sure what the result of that was. But we need to grow these things together to compare them. We only have very small seeds of Mignonensis at the moment, but I'm looking forward to getting them growing to the same sort of size. So it's a provisional name for something that hasn't got a name, basically. Very nice plant, actually, shrub.
Yes, it's growing very well in the botanic garden. However, because it has no scientific name yet, it's like waiting, but it's lovely, beautiful.
So the one unusual thing that happened with that last year was it was flowering very late in the autumn. Nothing else was flowering, and it developed fruits. That's strange because it's biennial. And those fruits seem to be coming true because there was nothing else flowering that could pollinate it. Oaks always do weird things.
Very cool. Thank you.
Yeah, thank you so much. Time for probably one more question. Feel free to unmute or put it in the chat. All right. Well, thank you so much again, Maricela and Alan. Really appreciate it. That was a wonderful presentation as well. And again, just a huge thank you to all of our presenters, Andrew Hipp, Maricela, Rodríguez, and Alan Coombes. Wonderful talks today. So we just greatly appreciate you taking the time to share your work with us. I know we have a few minutes left, so I'll pass off to you, Roderick, if there was anything you wanted to discuss before we wrap up.
Yeah, we don't have much time, but I do want to take the opportunity. I'm counting about 80 people we had, and this is like the biggest get together of IOS members in between conferences. So we were going to discuss several items. I don't know if we have time for many, but we announced our tour event. So if anyone has ideas or suggestions for places for tours or for visits for Oak Open Days, please do share, either speak up or put it in the chat or send us emails later. Certainly, we're always wanting more ideas about that. Also, ideas for increasing membership. We have a development and outreach committee that is trying to reach out and get more members. So any ideas of that would be welcome. I noticed Janine has a hand up.
Yes, Janine?
Yeah, Janine.
Okay, let's see if my connection holds out. My question was for Maricela about what happened after the hurricane to the oak trees, and are they doing okay, or did you lose any from that?
Oh, the trees in Acapulco, we don't know yet how they are. However, we know that all the large trees were fall down. The owner of and director of the Botanic Garden is really sad because they lost everything in trees. So even when they are now sprouting again, they lost that. So we expect that the quercus we send there, they should be okay because they weren't very big, but still we don't know the report. I just today heard that the gardeners, the Botanic Garden of Puebla sent to Acapulco to help them. As they say, is terrible, the conditions there. So they need help, really.
Thanks, Maricela.
Go ahead, Roderick. Were you gonna?
No, I was just going to say, if anyone has any of those comments on the topics I discussed earlier, love to hear from you. As I say, we're always waiting for new ideas, and I was also talking about our ideas of increasing memberships. We've had one of our goals for this year. Yes, Maricela.
Excuse me, just to complete the answer, sorry that I forgot. The rubramentas in Guerrero, they grow very far from the beach. So they grow in the mountain, high. So nothing happened in the mountains. Everything was in the coast, where the Botanic Garden we are working with is. Just that, sorry that I didn't say.
Thank you, Maricela.
Thanks, Maricela. Yeah, we'll be closing soon. I think we just have a couple of minutes left. So, yeah, Amy.
I was just going to mention too, I know, Roderick, we talked about with the iOS Instagram featuring people's photos of oak trees or landscapes that you have taken out in the fields or in your collections. So we would love to feature those on our Instagram, or feel free to tag the iOS in your posts and we can reshare them as well. So just if you have any photos you'd like to share with us to post directly, you can email myself or Roderick and we can get those up there. Or again, just tag the hashtag iOS or International Oak Society.
That's right.
I don't know if you want to put it in the chat, Roderick. I forget which one it is. And we'll be happy to reshare it just to kind of increase and highlight, the work that you guys are doing out in the field and all the cool stuff that you're seeing. I just wanted to mention that. Any comments or questions or ideas for future webinars or increasing iOS membership? Feel free to say it now or put it in the chat. Otherwise, you can contact us by email. We appreciate any and all feedback. Otherwise, was there anything else you wanted to mention, Roderick, before we wrap up?
No, thank you, Amy, for organizing this webinar and, in fact, getting the whole webinar action going. I think it's a great idea, and I think it's really worth doing. And I'm looking forward to the next one. One every 3 months sounds like a good schedule, and I hope we can keep to it.
Yeah,
So we need to find more people to present.
Yeah,
Volunteers welcome.
yeah, of course. So if you have any projects you want to share or work or your collections, feel free to email us as well, and we'd be happy to include you in the agenda for future webinars. But thank you, Roderick. Thank you, Maricela, Alan, and Andrew, and thank you all so much for joining. Really appreciate your time, and good to see you all, and thanks for being so interactive in the chat and during the discussions. I hope you have a great rest of your day, afternoon, and evening, everyone. We'll be in touch with the webinar recording. So thanks. Bye-bye.
Bye. Thank you all.